All right, so we're going to start the next chapter. So we're going to go into chapter 26, and we're going to discuss archaea and bacteria. So the objectives for this chapter are going to be to explain why bacteria and archaea are considered to be the most abundant, diverse, and ubiquitous organisms on Earth. We're going to describe the reproductive, morphological, and metabolic diversity among the prokaryotes. And we're also going to describe some key features of the major lineages of bacteria. Okay? So we're only going to focus on the lineages of bacteria. We're not going to go into archaea. So bacteria and archaea are two of the three domains of life. So if you recall, the third one is eukarya. That's the one that includes animals and also plants. Okay. So we're going to just focus on two of the three. Bacteria and archaea, these are mainly unicellular because, like I mentioned in the lab, there's always exceptions to these rules. But for the most part, they're unicellular. And they're also prokaryotic, so they lack a membrane-bound nucleus. But they also have differences between the two. So, for example, the types of molecules that you would find in the cell wall of bacteria would be different from archaea. And another example would be like if you introduced an antibody for a ribosome in bacteria, it wouldn't necessarily affect archaea and eukarya, yeah, because they are very different. So prokaryotes that live in your body are part of your human microbiome. A microbiome is a community of microbes that naturally inhabits a particular area. So it could be areas like your mouth, your hands, your intestine, and it also encompasses all the genetic material that's contained within it. So why do biologists study bacteria and archaea? Well, because of their biological impact. Okay. They're very abundant, so they exist in high numbers. Because of their habitat diversity, a lot of these are extremophiles, so they love extreme environments. Because of their medical importance, so we're going to go into Koch's postulates, which pretty much establishes a link between a pathogen and a disease. And that's going to lead us into germ theory, which accepts that pathogens cause diseases. And then we're also going to look at the role of bioremediation. So where you have microorganisms that you can use to get rid of pollution. So here's a table that shows some of the distinguishing features of bacteria and archaea. So as you can see here, there are differences, like if you're talking about the RNA polymerase of the two, there's differences in the peptidoglycan in the cell wall. So it's absent in one and present in the other. Then you also have differences in things like the first amino acid that's incorporated in translation. For bacteria, it's formylmethionine. For archaea, it's just methionine. So going into more of the biological impact. So bacteria and archaea are ancient, diverse, abundant, and ubiquitous lineages. So ubiquitous meaning they're found everywhere. So for example, the oldest fossils are of a 3.5 billion year old bacteria. And just to give you a perspective, eukaryotes weren't added into the fossil record until 1.75 billion years later. There's about 10,000 species of named and described, um, but there's hundreds, or certainly there's hundreds of thousands that it likely exist. There's about 1,000 microbes in the human large intestine alone, about 700 microbes in your mouth. So if you wanted to discover a new species, looking into bacteria and archaea would be the way to go because there's a lot to be learned in that field. So bacteria and archaea live almost everywhere. So from the Earth's surface to the Antarctic sea ice. Okay. And yeah, like I mentioned, recently entirely new phyla of bacteria and archaea have been discovered in the field of microbiology. Prokaryotes thrive in extreme environments. So that's why they call them extremophiles. They can live in places like hydrothermal vents, in very acidic environments where the pH is below 1, in very cold environments, uh, in very salty environments. So here you can see a picture where they're evaporating seawater, and you could see that after the water is evaporated, the salt crystals are left, and those are pigmented with archaeal cells. So they can tolerate uh, very salty habitats and environments. So extremophiles are a hot area of research because they can um, shed light into things like the origin of life. So they can help explain how life began on Earth. Astrobiologists also use extremophiles as model organisms in search of extraterrestrial life. And then there's also the commercial applications because enzymes that are found at extreme temperatures and pressures are used in industrial processes. So here's some bacteria that cause illness in humans. So remember, microorganisms that cause diseases are called pathogens. Okay. So we have things that you might recognize from the ones that we looked at in lab. So like, for example, Clostridium tetani, that's the species, and the disease that it causes is tetanus. So Clostridium tetani would be the pathogen. The disease or illness is tetanus. Another one that we looked at was chlamydia. So in this case, they're showing chlamydia trachomatis. So that would be the pathogen. And then the disease is the genital tract infection. Okay, so now we're going to go into Koch's postulates. So Koch's postulates, they show a causative link between a specific disease and a specific microbe. So in order for this to be established, there's some requirements that have to be met. So the first one is that the microbe is present in diseased individuals, but it's absent from healthy individuals. The next one is that the microbes must be isolated and grown in pure culture, okay, separate from the host. If the organisms from the pure culture are injected into healthy individuals, the symptoms should appear. And then finally, organisms that are isolated from the diseased experimental animal, again, grown in pure culture, should and demonstrated to be the same as the original organism. And then for germ theory, so Koch's experiments result were the first test of germ theory of disease. So it shows that some diseases are infectious, and it also goes into the process responsible for the pattern of transmission and growth of certain bacteria and viruses. So infectious diseases spread in three ways. So they're either passed from person to person, they're transmitted by bites from animals. Okay. So by the way, insects are animals. That's a typo here. So when they're transmitted by animals, those are called vectors. So, and then finally, they're acquired by ingesting contaminated food or water or exposure to microbes and surrounding environments. Okay. And so yeah, I think this is a good place to stop. So we'll pause here and then we'll continue on the next video.